Welcome back to another video on the channel where today we're going to be discussing lipids. So firstly we're going to look at the roles of lipids before going on to look at triglycerides and phospholipids, and then finally look at the test for lipids, the emulsification test. So let's jump straight in. Okay, so firstly let's discuss the roles of lipids. Firstly, lipids are a key source of energy for our bodies. When lipids are oxidized, they release a significant amount of energy, making them an essential component for powering various cellular activities, especially during long periods of exercise or fasting. Waterproofing. Lipids play a crucial role in waterproofing. They are insoluble in water because they are non-polar molecules. This characteristic is important for forming barriers that prevent water loss and also protect against water infiltration. This can be seen in the waxy cuticles of plants and the skin's natural oils. Thirdly, temperature regulation. Another important function of lipids is their role in maintaining body temperature. The fat stored in our bodies acts as an insulating layer, helping to retain heat and keep our internal temperature stable. This insulation is particularly vital in cold environments. Next, lipids also serve as a protective cushion around our delicate organs. Tissue or body fat surrounds and shields organs such as the kidneys and heart, which protects from physical shocks and injuries. Next, phospholipids, a type of lipids that are fundamental to cell membrane structure, contribute to the flexibility of cell membranes, allowing cells to maintain their shape and move. Additionally, they facilitate the transfer of substance into and out of cells, which is crucial for maintaining cellular function and communication. So to briefly summarize this slide, lipids are a versatile molecule with critical roles in energy storage, protection, insulation, and cellular function. Next, let's discuss triglycerides. So the structure. Triglycerides are composed of 3 fatty acid molecules bonded to a single glycerol molecule. These fatty acids and glycerol are linked through ester bonds, which form a crucial part of their structure. Fatty acids can vary in their bonding patterns. Saturated fatty acids have no double bonds between their carbon atoms, meaning all carbon atoms are fully saturated with hydrogen atoms. This structure allows them to pack tightly together. Monosaturated fatty acids contain one double bond between carbon atoms. This introduces a kink in the molecule, which affects how tightly they can pack together. And polysaturated fatty acids have multiple double bonds between carbon atoms, leading to even more bending and less dense packing. The presence of double bonds in fatty acids causes the molecule to bend. This bending means that triglycerides with more double bonds, such as polysaturated fats, cannot pack as closely together as saturated fats. This structural difference influences their physical state at room temperature and also their role in the body. Triglycerides are highly efficient energy storage molecules. They have a high energy content and a low mass to energy ratio. This means that they can store a lot of energy in a relatively small space, which makes them ideal for long-term energy storage. Triglycerides are also insoluble in water, which means their storage does not affect the water potential of cells. Additionally, when triglycerides are oxidized, they release water as a byproduct. This can serve as a vital source of water, especially for organisms in hot and arid environments. So to briefly summarize this slide, triglycerides are a versatile molecule with important roles in energy storage, structural function, and hydration. Their unique properties make them essential for various biological processes. Okay, so following on from triglycerides, we'll now talk about phospholipids. Phospholipids have a hydrophilic or a water-attracting head. This part of the molecule interacts favorably with water. In an aqueous environment, these hydrophilic heads face outwards towards the water inside and outside the cell. They also have a hydrophobic tail, which in contrast means it repels water. These tails orient themselves away from water and face inwards, away from the aqueous surroundings. This arrangement creates a barrier that helps to separate the internal environment of the cell from the external environment. When phospholipids are placed in water, they spontaneously arrange themselves into a bilayer. This bilayer forms because the hydrophilic heads interact with the water on either side, while the hydrophobic tails hide away from the water, aligning themselves in the middle of the bilayer. This structure is crucial to creating a stable and effective cell membrane. The hydrophilic phosphate heads of phospholipids not only help in forming the bilayer, but also contribute to the stability of the membrane surface. By interacting with the surrounding water, they keep the membrane intact and functional, ensuring that the cell maintains its integrity and regulates the movement of substances in and out of the cell. So to briefly summarize this slide on phospholipids, the unique properties of phospholipids, being the hydrophilic and hydrophobic parts, allows them to form a bilayer, which is fundamental to cell membrane structure and function. This arrangement is essential for maintaining cellular boundaries and facilitating various cellular processes. To wrap up this video on lipids, we will finally discuss how to test for them using the emulsion test. So firstly, we want to start by adding 2 centimeter cubes of our food sample to a test tube, and then adding 5 centimeter cubes of ethanol to the test tube. We will then dissolve the lipids by shaking the test tube thoroughly to ensure that any lipids present in the food are dissolved within the ethanol. This step extracts the lipids from the sample into the ethanol solution. We will then add water. After shaking, we will add 5 centimeter cubed of water to the test tube and shake the test tube gently again. After this, we should be able to observe the results. If a lipid is present, you will observe a cloudy white emulsion forming in the test tube. This cloudy appearance is due to the dispersion of lipid molecules in the water, indicating a positive result for lipids. So that concludes our video today on lipids, their roles, as well as discussing triglycerides, phospholipids, and then finally how to test for lipids using the emulsion test. If you enjoyed, feel free to like, comment and subscribe, share with your friends, tell them about the channel, and also feel free to check out any other videos on this channel on topics you might be struggling with for your A-level revision.